Distinguished ladies and gentlemen, I welcome all of us here to today's 409th inaugural lecture of our university. May I please give indulgence to please stand for the Vice Chancellor's procession in respect of this inaugural lecture. Thank you. Every living soul, please stand and remain standing. Kindly be seated, please. Let me welcome everyone to this 409th inaugural lecture series of Obafemi Awolowo University. The lecture for today is titled, All is Given, Food, Medicine, and More. To be presented by Professor Omolaja Osoniyi. Professor Omolaja Osoniyi holds a BSc Honors degree in Biochemistry from the University of Lagos, MSc 1992 and PhD 2005 degrees in Biochemistry from Obafemi Awolowo University, Ile-Ife. He had his postdoctoral studies in cancer biology at the University of Cape Town from 2007 to 2009. He joined the services of OAU in 1994 as an assistant lecturer and rose through the ranks to become professor in October 2020. Professor Osoniyi has benefited from various fellowships and awards from bodies such as the Federal Government of Nigeria, the International Atomic Energy Agency, Vienna, United Nations Conference on Trade and Development, and the South African National Research Fund, among others. Professor Omolaja Osoniyi has taught, conducted research, and published extensively in the life sciences. He has supervised numerous BSc final year projects, 16 MSc, and 9 PhD students. In his scholarly pursuits, Professor Osoniyi has delved into diverse research areas, ranging from the exploration of indigenous plants, ethnomedicinal properties, to the intricate relationship between food security and health through dietary interventions. He's a member of esteemed professional organizations such as the Nigerian Society of Biochemistry and Molecular Biology, the American Association for the Advancement of Science, and the International Natural Product Sciences Task Force. He has served the university in several capacities, including being coordinator of the pre-degree program in 2010, facilitator in the Directorate of Linkages and Sponsored Research from 2011 to 2014, and also as Deputy Director and then Director of the Central Science Lab between 2016 and 2020. He served on the Curriculum Development Committee of the African Union at the inception of Pan-African University Institute for Basic Sciences and Technology. Professor Osoniyi is happily married with lovely children. I hereby have the singular honor to invite Professor Osoniyi to present his inaugural lecture. Thank you. Welcome.
Vice Chancellor, sir. Principal officers, distinguished colleagues, guests of the university, ladies and gentlemen, welcome to the 409th inaugural lecture of Obafemi Awolowo University, Ile-Ife. It gives me great pleasure to deliver this, the 6th inaugural lecture from the Department of Biochemistry and Molecular Biology of this great university. I follow illustrious leaders like Professor Akintola Aboderin, who delivered his in 1981, Professor Afolayan in 1990, Professor Nojobi my PhD supervisor and mentor, delivered her lecture titled Fats and Life on 16th of February 1999, and the fourth lecture in the department followed in September 2019, delivered by Professor F.K. Agbola, and the most recent, titled An Odyssey into the World of Enzymes, was delivered by Professor Ayo Adewale on the 14th of March 2023. I'm grateful for the opportunity to deliver this lecture titled All is Given: Food, Medicine, and More. This title is inspired by scripture. In the words of John the Baptist, a man has nothing except it is first given to him from heaven. That's in John 3:27. These words are true on many levels for all of us, and even more so for a biochemist. The things we find out, the interactions we tease out at the molecular level, the benefits that accrue to mankind from our research endeavors, etc., are, in a sense, revealed to us, and sometimes serendipitously. Two quick analogies come to mind, inspired by the book, God, Technology, and the Christian Life. Think of children playing in a sandbox or with a Lego set. While the amount of sand in the box and the number of pieces in the set may be fixed, there's an infinite number of creations that could arise from them. The number and range of things created is limited only by the imagination and creativity of the children involved. We keep discovering many different aggregations of molecules and compounds. And different combinations of these compounds from nature are often put to use in different ways by different groups of people. Biochemistry studies the chemical basis of life. It is at the very core of the life sciences. It began essentially by applying the tools of chemistry to study the molecules involved in maintaining growth and other life processes. While life without contributions of biochemistry is almost inconceivable, we are also at the center of every controversial discovery in the life sciences. We are at the forefront of playing God, as science skeptics posit. The name biochemistry was coined by Carl Neuber in 1903. So our subject is more than a century old. Unsurprisingly, much of the early work focused on an attempt to understand the fundamental macromolecules, proteins, nucleic acids, carbohydrates, and lipids. Work by Oswald Avery et al. In the 1940s provided clear indication that deoxyribonucleic acid was the genetic material of the cell. The structure of DNA was elucidated by Francis Crick and James Watson and published in 1953. Their proposed structure suggested the way in which genes might be replicated. Not long after, Francis Crick linked the sequence of bases to the amino acid sequence of proteins, and the amino acid sequence in itself determined the three-dimensional structure of these proteins. By 1960, it was established that a sequence of 3 DNA bases encodes an individual amino acid. The sequencing of significant lengths of DNA remained a difficulty until 1977. But by the mid-1980s, a dream came up to sequence the human genome, and the task was completed in 2003 with a draft sequence published in 2001. Parallel to the DNA sequencing efforts was the use of restriction enzymes, which cut DNA at specific points in the chain, allowing us to get to the point of recombinant DNA technology, transgenic technology, and other tools that gave rise to the rapid development in the 1990s of biotechnology based on gene splicing. The era of man playing God had finally arrived, because we could finally accomplish genetic modification of living organisms. Every aspect of life as we live it on this planet now has major contributions from biochemistry. We have enhanced everything from animal production to plant production to health and even management of environmental resources. However, our good intentions have not always translated to the general good. There are real and perceived problems, and there's often a gap between intent and application of knowledge in science. Even well-intentioned biochemical research can have unforeseen negative impacts if ethical, legal, social, and environmental implications and consequences are not thoroughly evaluated. The current debates in the public space about genetic medicine and genetically modified organisms are evidence of this. Uncontrolled and irresponsible use of biochemical technologies will have bad consequences for us all. We must therefore consider the ethical implications and potential impacts of our work on society and the environment. The governance and ethics frameworks considering what is best for society must be strengthened. How did I get here? I enrolled for a degree in biochemistry at the University of Lagos. My BSc final year project supervisor, Professor Owe Magbagbiola, sealed my love for the subject because she was a wonderful supervisor to our cohort. And beyond just reporting results of experiments, we discovered a love of research and we were inducted into the world of weekends in laboratories. By a series of divinely orchestrated happenstances, I found myself in Obafemi Awolowo University, Ile-Ife, for postgraduate training in Hamatan semester 1989-1990, and ended up in the laboratory of a woman with a titanic reputation, Professor F.D. Onojobi. She had a long-standing interest in lipids generally, and particularly eicosanoids. She had worked on prostaglandins and their synthesis in plants. Prostaglandins are hormone-like mediators in many physiological and pathological processes, and they are found in vertebrates and in some terrestrial and aquatic invertebrates. They and their related eicosanoids were rarely found in plants. However, they had been identified in some plant species around 1973-74, first discovered in onions and later other plants. The direct precursors of prostaglandins, such as arachidonic acid, dihomogamma linolenic acid, eicosapentaenoic acid, are also found in various plants and algae. Their precise functions in plants are still not fully understood, but research suggests they can influence plant processes such as flowering, fruit ripening, and mediation of stress responses. A full understanding of their role is still a matter of ongoing scientific investigation. About the time I was considering a research project, I came to know about an ephedrine soup that was prepared for women to ease labor and promote afterbirth recovery. And this fascinated me. Ephedrine is a common medicinal plant, and it was a major interest in the Onojobi lab. And I therefore joyfully joined my colleagues Akinfa Murewa and Emmanuel Ironge of blessed memory, who were working on different ephedrine projects in the Onojobi laboratory. The lab had previously demonstrated the presence of prostaglandin-like compounds in ephedrine. My project used radiolabeled arachidonic acid, a known substrate for prostaglandin synthesis, to probe the factors affecting the synthesis of prostaglandin-like compounds in Ocimum gratissimum. I was able to demonstrate the conversion of arachidonic acid to prostaglandin-like compounds, and I was also able to work out the optimal conditions for the synthesis. There were two distinct pH optima, one similar to that of animal prostaglandin synthesis at between pH 7.5 and 8, and the other at pH 6.5. These were novel findings, and they suggested that there were different mechanisms for prostaglandin synthesis between plants and animals. I returned in 1994 to join the staff and began a PhD, but this time my attention was on another plant, Jatropha coccus, known as lakpa or botuje. At the time, it was one of the most researched plants in the world for various reasons. The use of medicinal plants as hemostatics is an age-long practice across cultures. In many tropical regions, the latex or sap of Jatropha is a well-known traditional remedy used for this very purpose. When applied to a cut, it is known to help stanch the flow of blood. For generations, its role was simple and undisputed. But I decided to take a closer look and hopefully isolate the compounds responsible for this bioactivity. Our first results quickly showed that the active principles were extractable into organic solvents. We also found that the same plant sap used to stop bleeding also showed anticoagulant properties under certain conditions. When the whole undiluted latex was tested, it performed as expected based on traditional use. But when the latex was diluted, it began to show the opposite effect, actually prolonging the clotting time so much that the blood did not clot at all. This was the first documented report of the anticoagulant properties of Jatropha coccus. Attempting to resolve that paradox became a major part of the remaining work for the PhD project, and we needed a collaborator who would help us. I was fortunate to come in contact with Professor A.O. Ogundaini of the Department of Pharmaceutical Chemistry, who I'm pleased to say is here today. Thank you, sir, for helping me out then and continuing to help me out even now. It's good to honor your supervisors. So he was supervising my friend then, and I was learning vicariously through my friend from him. And when it was time, I discussed with Professor Nojobi, and we discussed with him, and he readily agreed. And he has been with us since. Now, eventually, I was able to use solvent partitioning to resolve the activities and resolved the coagulant activity into ethyl acetate, and a butanol fraction contained the anticoagulant activity. This paradox with Jatropha coccus latex exemplifies the layers of complexity that exist in natural products research. We're reminded that a single traditional remedy can possess dual or even paradoxical functions that are only revealed under serious scientific scrutiny. And this also explains partially the fact that a single plant may be used to treat different conditions in different places. Each people group evolved the processing and dosage that works in their specific instance for their chosen purpose. And the way natural remedy is prepared and used can be as important as the remedy itself. My introduction to cancer biology. I applied for and secured a postdoctoral position in the University of Fort Hare in South Africa. And I was to work in natural products chemistry beginning from January 2007. While getting ready to resume there, another opportunity came up, and I secured a 3-month advanced laboratory training fellowship to the University of Cape Town, sponsored by UNCTAD. And it was to hold from October 16 to January 16, 2007. I was assigned to the laboratory of the program director, Professor Iqbal Parker, in the Division of Medical Biochemistry at UCT. It was a cancer biology lab, and his group included Denver Hendricks and a few others. I told them of my plan to proceed to Fort Hare in January when the program finished, and I wrote to my prospective host in Fort Hare, notifying him of these developments and to secure his permission to resume in mid-January. But by December, I was offered another postdoc position in UCT. Apparently, the cancer biology group did not want me to go, and I also chose to accept. My direct supervisor was Denver Hendricks. And it was a steep learning curve for me, learning the rudiments of cancer biology and the biochemical techniques needed to confirm activity and work out mechanisms of action for the compounds that our collaborators were synthesizing all across South Africa. According to the IARC Global Cancer Observatory, nearly 1 in 6 deaths were caused by cancer in 2022. And the number of new cases in Africa is more than 1 million, resulting in more than 700,000 deaths. Many cancers can be cured if detected early and treated effectively. But therein lies the problem. It's neither detected early nor treated well enough for various reasons. Some of the most common cancer types, such as breast cancer, cervical cancer, oral, colorectal, have high cure probabilities when detected early and treated according to best practices. And there is, however, a significant variation in treatment outcomes between countries of different income levels. 90% of high-income countries have good success, less than 15% success in low-income countries. So there is therefore a need to keep working on finding newer, better, and more affordable drugs and treatments. So the major research interest of the Hendricks lab was esophageal cancer, which was a problem in South Africa because they had a 5-fold higher risk of developing the cancer than the world average. And so they invested in it. That was the environment that I found myself in the Parker Hendricks lab. And our collaborators in UCT Chemistry Department, Rhodes University, University of the Western Cape, and a few other universities were all united in the quest for finding better anti-cancer agents. The strategy was simple. Bioactive compounds were extracted from various sources, and synthetic analogs were made as directed by the results of our biological testing of those compounds on cancer cell lines. That was done in our laboratory, and a lot of that was done by me while I was there. Many of the significant findings have resulted in promising compounds. In one study, we synthesized 4 2-substituted 1,4-naphthoquinones related to the marine natural product 2-deoxylapacol, a compound that had previously been shown to be active against squamous esophageal cell cancer. We assessed cytotoxicity using various assays, and we were able to show that our 4 synthetic compounds were better or comparable to the currently used drug at the time, cisplatin. In a similar study, we investigated extracts of Trimusculus costatus, one mollusk from the South African coastline, and developed 2 new labdane ditapines and 3 new metabolites, which were also screened against human esophageal cancer cell lines, WHCO1. And again, the activities were comparable or better than that of cisplatin. In more comprehensive structure-activity relationship studies, we revisited the previously mentioned naphthoquinones and developed a series of 25 related synthetic compounds. And we screened against WHCO1, and most of the compounds, when compared to cisplatin, were either good or better. The mechanism of cell death on treatment with these compounds was also worked out. And for one of them, we were able to show that the Janus kinase C-Jun activation pathway was the mechanism. And this happens to be the first description of that pathway activated by naphthoquinone compounds. Sir Vice Chancellor, sir, I returned to Ile-Ife after postdoc, and it was a traumatic experience because there were technical hitches with every cancer project designed, and simply for lack of resources. So I had to go back to the drawing board. I went back to nutraceuticals. My first MSc student, Omede Ame, was transferred to a colleague when I was starting my postdoc. And he had just started bench work before I left. So we had designed the project, everything was all set for go. And it was sad for me to find him still on the bench when I returned. He was bright and hardworking, so I couldn't explain the delay. And so my first priority was to see him complete his work and defend his thesis. And in this was also my redemption, because I had no choice but to get back on the bench. And the project was simple, straightforward, and we were able to make it work. A nutraceutical is any substance that may be considered a food or part of a food that provides health or medical benefits, including prevention and treatment of disease. And our foods are indeed medicinal treasure troves. So Ame's project was to evaluate the lipid-lowering effect of Moringa oleifera using a detergent-induced hyperlipidemic model. And we found that ethyl acetate extracts of Moringa mitigated all the effects of dyslipidemia occasioned by the detergent. Beyond confirming the health benefits of Moringa, this model became one of our primary tools for probing the efficacy of other plants and condiments reputed to have lipid-lowering properties. And the methodology has since been adopted and used very effectively. Another study in the lab established antioxidant and anti-hyperlipidemic properties of iru, or dawadawa, a fermented condiment prepared from the seeds of Pacea biglobosa, and ogiri, another fermented condiment prepared from the seeds of Citrullus vulgaris. That was my second MSc student, and eventually she became my first PhD student. Globally, fermented foods form an integral part of the staple diet of people. And Iru and Ogiri are used all across West Africa. In addition to their role in the cuisine, there are various claims about their efficacy in treating various conditions. All indices of dyslipidemia and oxidative damage were mitigated by a dietary inclusion of Iru and Ogiri in a dose-dependent manner. And we found also that a histological examination of the liver of our test animals suggested that the condiments also possessed hepatoprotective potential. And the results imply that their consumption may be a possible dietary measure for the control of hyperlipidemia. Essentially, the same methodology was used to probe the hypolipidemic properties of some other foods. And similar studies conducted by the group on aridon, which is Tetrapelura tetraptera, a plant used extensively in the fattening room practices of the south of Nigeria, proved that that plant was also a hypolipidemic agent, or had hypolipidemic agents. We similarly found the same with Solanum nigrum, a fodu, which Yorubas eat quite well. Beyond lipid-lowering properties, we investigated other bioactivities in some other vegetables consumed as food. And one of the other vegetables we studied was Crassophyllum crepidioides, or Efoebolo, commonly known as ragweed or fireweed ragleaf. In many African countries, it is used as a valued vegetable. In Okoyemi Oyedele's PhD project that was co-supervised by my PhD supervisor, Professor Onojobi, and I, we conducted a series of studies on this plant. And we found that ebolo, the methanol extracts and fractions, had significant anticoagulant activities which may be exploited in the treatment of blood coagulation disorders. With the results of our in vitro experiments, we further decided to see if the plant could produce similar effects in a living organism in a diabetes model. We found that ebolo and its fractions significantly reduced plasma glucose levels and mitigated damage due to the damage to the pancreas, which is the organ responsible for producing insulin. And the study suggests that the plant may help regenerate these vital insulin-producing cells of the pancreas. The extracts also significantly help to restore a healthier lipid balance and atherogenic indices. Analysis of the hexane fraction, which was particularly effective in our rat study by GCMS, revealed several bioactive compounds known for their bioactivities, providing a direct chemical explanation for many of the bioactivities our study revealed. As I said earlier, Ayolawal returned to my lab for PhD studies, and we decided to have a closer look at Iru and Ogiri. The studies were part funded by a fellowship from TWAS, which enabled her to do a research visit to one of our collaborators in South Africa, where she was able to do parts of the work that we could not do here. And this time, our focus was on antimitotic, antiproliferative, and anti-cancer properties of the extracts of these fermented foods. And our studies, for the first time, showed that extracts from Iru induced time and dose-dependent cytotoxicity in all tested cancer cell lines, breast, cervical, and liver cancer. Notably, higher concentrations were required to inhibit the growth of normal KMST6 cells, indicating a selective action against cancer cells. Furthermore, treatment with the aqueous extract caused cancer cells to lose their original structure and detach from culture plates, indicating that it was a very effective agent, actually even reducing the ability to reproduce and form colonies in the cells. The mechanism of action was also determined. And similar to Iru, Ogiri displayed the same properties. And one interesting thing with the Ogiri was that the KMST6 cells appeared to recover from exposure to the extract after 48 hours. And this was beautiful because it means that the plant will kill cancer cells and normal cells will recover. The collective findings from these studies robustly demonstrate that traditional West African condiments possess significant anti-cancer properties. And both condiments exhibit dose and time-dependent cytotoxicity. And the studies also established a strong foundation for the potential of those sources, those compounds, or those foods as sources of nutraceuticals with anti-cancer properties. The periwinkle projects. Sir Vice Chancellor, sir? Periwinkles are almost a staple in the cuisine of the people of the South-South. Some of us have also grown to enjoy them. They are marine mollusks, and the two most common of the eating species are T. fuscatus and P. aurita. And they are also found all across the coast of West Africa, Angola, and Gabon. And notably, T. fuscatus is utilized in the traditional folk medicine of Nigeria's South-South. This is not surprising because their big cousin, the African land snail, is well known in the ethnomedicine of West Africa. The general consensus among researchers in the field is that traditional knowledge of coastal and indigenous communities who have used mollusks for food and medicine could provide invaluable leads for targeted scientific investigation. And this was the basis of our probing this plant, and our student, Queensley Ehianruwa, took on this project for a PhD, which I co-supervised with Kenyan collaborators at PAUSTI in JKUAT, Kenya. And it was followed a little later by projects in the lab here for Paul Abraham and Grace Okmaride for their MSc projects. The projects were funded by JICA and the AU through PAUSTI. Crude extracts and their peptide fractions from both mollusks were tested for a wide range of biological activities across several in vitro and in vivo models. And the main findings are that T. fuscatus extracts showed significant activity, killing cells by the preferred apoptotic method, and significantly upregulated key pro-apoptotic genes, P53, caspase 3, and caspase 8. Our studies also confirmed that the periwinkles, the extracts, possess significant anti-inflammatory and peripheral analgesic properties, supporting their traditional use in treating wounds. Both alcohol and aqueous extracts from the two species significantly inhibited production of key pro-inflammatory mediators. Protein hydrolysates derived from the periwinkles by simulated gastrointestinal digestion were found to possess potent angiotensin-1 converting enzyme inhibitory activity. And this is important because ACE is a key target in antihypertensive treatments. And we found this activity was significant in our periwinkle extracts. Antimicrobial potential of the extracts were also evaluated. And using various methods, we were able to show activity, indeed bactericidal activity, against 5 susceptible bacterial isolates, and bacteriostatic activity against Candida albicans, a fungus. Several analytical techniques were used to isolate the compounds that were responsible for these activities. We extended the mollusk study to investigate antioxidants, cytotoxic, and antibiofilm activities of crude extracts of another edible marine mollusk, Thais coronata. That was Grace Okmaride's project, which we are still working on with Professor Oiruka Okeke's lab in the University of Ibadan. Similar to what was obtained for the other periwinkles, the results of the assays indicates that we have significant antibiofilm activity, which could protect cells from all kinds of bacterial attack. Taken together, these mollusk studies demonstrate that the whole body extracts of T. fuscatus, P. aurita, and Thais coronata are rich sources of bioactive compounds with multifaceted therapeutic potentials. And these studies also provide a strong scientific validation for the traditional medicinal use of these mollusks. Now on to fishy business. Many nutritional benefits attributed to fish are particularly obtained from their advantageous fatty acid profile. In recent years, significant attention has been focused on the polyunsaturated fatty acids, and they are practically, the important ones, EPA and DHA, are practically found only in fish. And so, before we got in, most of the published work on the nutritional value of our fishes focused primarily on proximate analysis at the time. We chose to focus on the importance of the lipids. And so we did a comparative study on Clarias garipinus, looking at wild and cultured catfish, and to see how the lipid profile affected the quality of the fish. While proximate analysis showed there was no significant difference between the wild and cultured fish, we found that the wild fish demonstrated superior levels of beneficial polyunsaturated fatty acids. In a second related study, we look at another problem related to fish. Tilapias are practically second to Clarias in terms of popularity with fish lovers and aquaculturists. But the problem with tilapias is that they are not as hardy as the aquaculturists would have them. So we decided to look to compare the wild tilapias with cultured tilapias. And one of the critical findings was that a unique single nucleotide polymorphism in the freshwater Coptodon zili was important because it resulted in a truncation of the TNF-alpha protein. And we propose that this is perhaps what is responsible for the hardiness of the wild fish. This genetic diversity has implications because it might be a potential resource for improving farmed fish stock. Studies on Abrus picatorius. Mr. Vice Chancellor, sir? Professor Nojobi and I co-supervised another student who looked at Abrus picatorius, and he was also fortunate to get an ICCBS TWAS fellowship for a research institute, for a research visit to the University of Karachi. And it was truly divine intervention because we were stuck before he got that fellowship. And ultimately, we discovered a rare class of isoflavonones known as the abroquinones in this plant from the root. 7 of them were previously known, but 4 were previously undescribed. So we were the first to describe them. The fractions and isolated compounds from Abrus picatorius demonstrated significant and selective cytotoxic activity against a range of human cancer cell lines. The plants and their extracts also showed a broad spectrum of activity against protozoans, fungal, and bacterial pathogens. And a significant one was their activity against Leishmania, Leishmania major and Leishmania tropica. And it was the first time that abroquinone A and B were shown to be active against this pathogen. We were also able to show that our compounds were very active against multidrug resistant Staph aureus. The compounds, in addition, exhibited potent anti-inflammatory activity by modulating key compounds or key components of the immune system. In pathogenic bacteria, urease is a virulent factor that's essential for the colonization of host organisms and maintenance of bacterial cells in tissues. And research geared towards identifying potent urease inhibitors is important. So we are happy to find that some of our compounds, particularly abroquinone A and B, were very active against urease. White death and Garcinia cola. White death is the nickname for TB or tuberculosis. And it has always been a major health burden. And multi-drug resistant forms have emerged in the era of HIV. Standard treatment is a multi-drug long treatment program for a minimum of 6 months. Standard combination of isoniazid and rifampicin for 6 months is the common cornerstone of this therapy. While effective in 90% of cases, there's often failure because patients are often sicker from the drug than from the TB, and therefore they stop taking it, and there's also adverse effects with neurotoxicity and hepatotoxicity. So we looked at the ability of Garcinia cola or common orogbo to protect and mitigate some of these adverse effects of the INH and rifampicin. And we're able to report that, yes, there's subclinical neurotoxicity in the administration of this combination, and that the use of Garcinia cola extracts mitigated this damage, including the damage done to the liver. This was subject of the investigation of one of my MSc students, Ogono. Biological approach to weed and pest control, bioherbicides and bioinsecticides. Modern agriculture depends a lot on chemical inputs like fertilizers, herbicides, and pesticides. An accumulation of those chemicals in the environment is an ongoing concern. So it's necessary to develop bioherbicides that will be biodegradable and environment friendly. And this informed a series of studies in our lab in the period from 2011 to 2016. And we studied extracts of various plants. Dioscorea dumetorum, Costus afer, Hilaria latifolia. And we explored allelopathy, genotoxicity, and cytotoxicity. And all our studies met with good success because we found a significant number of extracts that could act as bioherbicides. And of course, they are biodegradable, meaning they are better for the environment than synthetic herbicides. It is common to see the insect pests as the second half of the problem. And so we also looked at developing biopesticides. And we chose the housefly as our model. And we hypothesized that if we could make the flies weaker and ultimately shorten their lifespan by tampering with their diet, we could make them ineffective pests over time. And that's precisely what we did by tinkering with their base diet and making it fat-rich. And we significantly reduced their lifespan from 17 days to about 11 days. This study points to the fact that we can use this simple trick to bait insects and make them ineffective pests. Lassa virus studies. Adetunji Adesina and our collaborator, Professor Ayolayemi, and a few others across southwestern Nigeria were involved with the studies of Lassa fever. We all know Lassa fever is Ibaore, and there's almost a yearly attack or outbreak of Lassa fever in this country. we studied the Edo and Ondo axis, and we found a series of important things. The originally previously identified reservoir for the virus is Mastomys natalensis, and it's ubiquitous across the region. And it would have been okay to just assume that that is it. But in addition, we found that, one, the Edo-Ondo region exhibits exceptionally high rates of active Lassa virus infection in both that primary rodent reservoir, but two, that Mastomys erythroleucos is also now an active reservoir for Lassa virus lineage 2 within that axis, co-circulating with Mastomys natalensis. 3, we found that rodent abundance, not prevalence, drives human risk because Lassa virus prevalence in rodents is consistently high year-round. The primary driver of human Lassa fever outbreaks is the seasonal peak in indoor rodent abundance. We found also that urban centers were hubs for viral dissemination. Counterintuitively, time-calibrated phylogenetic analysis consistently show that Lassa virus sequences derived from humans in key hotspots like Ecoma are ancestral to those found in local rodent populations. And this finding raises the possibility of a more complex bidirectional transmission cycle that includes reverse zoonosis. In other words, human to rodent transmission. 6, time-calibrated phylogenetic analysis of rodent-derived sequences also indicates that Lassa virus variants in Edo State are ancestral to those in the more westerly Ondo State, supporting a broader east to west migration pattern of the virus across southern Nigeria. The continuous spillover of Lassa or Lassa virus into abundant commensal non-reservoir species poses a significant long-term risk of the virus adapting to a new host. These findings bring a better understanding of Lassa virus ecology and transmission dynamics in Nigeria, and hopefully will help to shape a better response by the health authorities to Lassa outbreaks in Nigeria. Evaluation of Sphazodia campanulata exudate for anti-cataract activity. Cataract, characterized by opacification of the crystalline eye lens, is the world's leading cause of blindness, with a disproportionately high incidence in developing nations. Sphazodia campanulata is the African tulip tree, widely used in traditional medicine. It's called mojutoro. Some people will identify that it's just outside Oduduwa Hall here. And that name simply means something that clears the eye, mojutoro. And the exudate from the buds is commonly applied to the eyes as a local remedy. We therefore decided to evaluate the anti-cataract activity of Sphazodia campanulata flower bud exudate because there was no previous study before ours. The study employed an in vitro organ culture model using lenses harvested from healthy white albino rats. And morphological assessment of efficacy was done after treatment for 72 hours. And the lenses were photographed on a grid to visually evaluate the opacity and transparency of the lenses. We have to thank Professor Falui especially for his help in this regard, because he had to develop a whole range of new photographic techniques for us for the study. We were able to confirm potent, dose-dependent, effective protection of the lenses by Sphazodia campanulata exudate against galactose-induced cataractogenesis. The study also provided that scientific validation for the traditional use of Sphazodia campanulata exudates in Nigerian ethnomedicine for improving vision and treating eye disorders. Complementary and winning foods. the Grow Star project. Complementary food is required for healthy nutrition in infants and young children from age 6 to 24 months. By this time, breast milk is no longer adequate for optimum metabolic support. And commercially produced complementary foods are expensive. And the locally formulated complementary foods are either poorly formulated or inappropriately processed. And so the Grow Star project was initiated to develop a cereal legume-based complementary food that is formulated and processed to meet the nutritional need of tropical infants and young children. The project resulted in 9 different complementary foods with very dynamic and novel nutritional features, which were collectively registered as Grow Star, a winning food for tropical children. The novelty of this product attracted an utility patent from the Federal Republic of Nigeria. Grow Star contains adequate macronutrients and minerals, and with very low anti-nutrients. And its profile is fantastic for growing children. It's more affordable than most of the branded products on the market, has low moisture levels compared to related products, therefore guaranteeing longer shelf life, and does not tolerate microbial invasion. We evaluated Grow Star using the Canadian Protocol of Evaluation, and it meets the recommended standards. It's easy to prepare and store and does not contain any mycotoxin associated with poor management of grains against other local products. The project resulted in the award of a PhD for Dr. Ama Gongonte, who is now a senior lecturer in Babcock University. There are other projects that we also participated in with serious significance, including one involved in managing sickle cell anemia by manipulation of the diet with significant success. Ojo, Osoniyi and Aboderi, and Ojo et al. 2006. We also looked at many other plants, especially those involved in malaria. Mr. Vice Chancellor, sir, permit me to talk about a few other contributions. I have had the opportunity to contribute to teaching and service at every level of this university and the society at large. I have served the Faculty of Science and the university in various standing and ad hoc committees, and I have served Center for Distance Learning, the Directorate of Linkages and Sponsored Research, CATREC, the Central Science Lab, and many other committees of this university. We have also served the African Union and the FAO in various capacities. Mr. Vice Chancellor, sir, permit me to make this recommendation. Many natural product researchers like myself come up with a lot of interesting lead products that could be developed into bioherbicides, biopesticides, nutraceuticals, and medicines. I think it is high time that the university developed a consistent and intentional mechanism for investing in the raw products of our research to the point of marketable products, and to be a huge source of revenue for both the university and the researchers and the society at large. Mr. Vice Chancellor, sir, in closing, I believe that God is not waiting to be surprised by what we might discover in our research. On the contrary, he allows us like those proverbial children playing in their sandboxes, to come up with interesting and beneficial things. It's up to us to strengthen our ethical guardrails and regulatory frameworks so that we will play God in a positive way for humanity. In my research, I have had the privilege of discovering beautiful things concerning food, medicine, and much more, as you have seen. It has been a pleasure to share some of that with you here today. In the time left for us to contribute to research and teaching and service to humanity here, we are ready to keep asking and to receive much more. Acknowledgements and appreciation. I appreciate the contributions of all my teachers, some of whom are seated here, starting from primary school to the present. I also sincerely thank the students I have had the honor of interacting with since I joined the university. They have provided significant discussions that led to asking the question, what if, and so what? And then we probed, and we found things. I also thank my colleagues for stimulating and vigorous discussions. I have been particularly blessed by the insight and generosity of my collaborators here in this university and across the world. I'm also grateful for all the technical staff who have helped me over time. The data works because you worked. I'm especially grateful to my faith family, the Redeemed Christian Church of God, here in Ile-Ife and in Cape Town and Stellenbosch in South Africa. I'm also grateful to the Nigeria Christian Graduate Fellowship. Thank you for being there for me. Thank you for being there for my family all these years. Thank you for being there even till today. I'm grateful to my parents of blessed memory who looked beyond their own circumstances to a better future for my siblings and I. I thank God for their commitment, dedication, and sacrifice. I thank God for my siblings and their spouses who have been a blessing to us. I'm also grateful for my other parents who unfortunately could not be here today. They're very old people. They have supported my family in every way imaginable throughout this journey. I'm equally grateful to all the Ogunjurans and their spouses. Thank you. I sincerely thank my wife, Olayinka Oluwafunmike, who has sacrificed more than many can know to strengthen us and support our family. My children, Joshua and Oluwase Ugba, Oluwase Ilayo, Ini Oluwa, Oluwafirewamiri, who have always known and understood that Daddy had to be in the lab, working on one project or the other, before they even started school themselves. I also thank my children in the BSCIF. Thank you for enriching my life beyond words. And finally, I give glory to God. Thank you, God, for allowing me to be here, to partake of the wonders of your world, to dream and to play in your sandboxes and to discover things of beauty and joy. Thank you for the privilege you have given me to share a bit of that joy and beauty with the people in my world, your world. Sir Vice Chancellor, sir, ladies and gentlemen, friends, I thank you for your presence.
If it had not been for your mercy, oh Lord,
Certificate presentation. Obafemi University, Ile-Ife. Certificate of presentation of inaugural lecture. This satisfies that Omolaja Osoniyi, a professor of biochemistry in the Department of Biochemistry, Faculty of Science, Obafemi University, Ile-Ife, Nigeria, delivered the 409th inaugural lecture titled All is Given, Food, Medicine, and More. This day, November 11th, 2025. Congratulations.
Let's go. Woo. Somebody lift up a shout. If it had not been for your grace, or a fashion bound. If it had not been for your mercy, I know shining Morida. And you say, if it had not been for your grace, or a fashion, if it had not been for your mercy, I know shining. Say it again, say, if it had not been for your grace, for your fashion, if it had not been for your mercy, say I know shine. Let's go. So somebody say grace, grace, for your fashion. Somebody say mercy, mercy. I know the more. Say grace, grace, for your fashion. Somebody say mercy, mercy. Hey, let's go. Somebody lift up a shout if you have received His grace. If it hadn't been for your grace, yes, sir, or a fashion,